we start? So can you hear me? Am I audible?
Yes, ma'am.
So can we start the next session? So can we start? Professor Trivedi, sir, can we start? Welcome back, sir. Uh, Sir has already joined with us, so we will be starting our next session where Sir will enlighten us with the practical part of the topic. Thank you, sir.
Hello everyone, welcome back. Sorry. Hello. So welcome back after the lunch. So let's start. These practical videos are pretty long. Okay. So what I will do is I'll share these videos, that the links, so that you can be able to watch them as per your convenience, and you can watch it now, you can watch it later, right? So just after this talk, maybe I will share the links with the organizers and they will share with you. What we are going to talk about is more into the advanced stage, right? Because now what we have done is we have isolated the gene, right? And uh, once you isolate the gene, your next objective would be that you are going to supposed to clone that gene into the suitable vector, right? And so that you can be able to use them for downstream applications. So in this particular session, what we are going to do is we are going to discuss about how you can be able to clone into the suitable vector, right? And then how you can be able to use that particular clone for protein production. So I'm sharing my screen. Is it visible now?
Yes,
Yes,
sir.
sir.
Yes.
Okay, good. Okay, so sorry. So this I think we have already discussed, right? So in this particular scheme of molecular cloning, two components are very important. One is this, right? From where you are actually going to extract the gene of your interest. It could be either by the genomic library preparations or the cDNA library preparations, or whether you use the PCR and get this particular fragment and ultimately all that, right? So, and then the second is your transforming agents. So, and then once you got this, the ligated plasmid, then you have to transform this into the host. So basically, in a downstream to the cloning, right, you always require the two components, or you require the two, the two parts actually. Okay, one is your, the recombinant clone, right, and the second is your host, right. Now, talking about and these two are compatible to each other, right? For example, right, in a two component system, right, you basically require the host cells, right, or you actually require the transforming agents. Okay, host cells could be prokaryotic in nature, for example, the bacteria, or it could be eukaryotic in nature, so that such as the unicellular eukaryotes such as yeast. It could be multicellular animals, or it could be very very complicated plants. Similarly, you can also have the transforming agents. I have taken an example of the plasmid that is belonging to the prokaryotic system, but it could be another other kind of the vectors. You can have the vectors which is only propagating into the yeast, such as the yak, or you can actually have the mammalian vectors, right? Or you can also have the plant vectors. So this two component system is very very important, and it is very difficult, important for you to understand also that the two component system, both of these, the transforming agent and as well as the host, has to be compatible with each other. Okay, it's not like you are taking a plasmid and putting into the plant system, or you're taking a, the yeast artificial vectors and putting into the animals or plants, right? Because that will not grow, because these transforming agents do have the certain requirements which only be met in their compatible host system. So transforming agents and the host are supposed to be compatible with each other. Now, talking about the host, what is host? The cell which is actually being used to propagate the particular type of transforming agents, right? So, in a biotechnology, a host organism serves as a platform for producing the particular compound, such as protein or the metabolite, or it is utilized to replicate and express the foreign genetic informations. So, in host, as far as the host organism is concerned, we can have the host like from the yeast. You can have the mammalian cells, plants, bacteria, or the in the bacteria such as the Bacillus subtilis and the E. coli. Right? Bacillus subtilis is recently very popular in terms of producing the different types of proteins because of the simple reason that E. coli is very simple system, but E. coli does have the problem of the codon biasness and so on. And then the folding machinery of the E. coli is also very different when you are trying to overexpress a protein from the mammalian system or the yeast system. So that's why the Bacillus subtilis is becoming more and more popular in terms of the choice for overexpressing a particular protein which is from the eukaryotic system. Then host should allow the easy entry of the recombinant DNA into the cell and should not consider the recombinant DNA as a foreign DNA and degrade it. Remember that every organisms do have their defense system, right? So the host cells are actually having a defense system, right? And this defense system recognizes who is belonging to them or who is belonging to the outsiders, right? Remember like everywhere, right? Whether if even you when you are staying in your home, right? You know which one is your family member and which one is your enemy, which one is your externals, right? So similarly the host cells are also like that, right? If the you are talking about the E. coli, right? E. coli knows which DNA is belonging to themselves and which DNA is belonging to virus, which DNA is belonging to animals, and so on. So host cells are being conditioned in such a way that it allows the entry of this foreign DNA and it does not recognize that DNA as a foreign DNA. Okay. And then the second part is the host must supply all the required enzymes, right? It should supply the RNA polymerase, it should supply the DNA polymerase, it should supply the ribosomes, and so on, right? So that you can be able to use that for protein production and allow the replication of the vector DNA along with the inserts. Now, talking about who can be hosts, so we have taken an examples of the prokaryotic system. We have taken an example of the yeast. We have taken examples of the animals and as well as the plants. Now, talking about the simple system, so you can actually be able to use the E. coli as the host, right? So E. coli is a gram-negative bacteria, right? And it is very, very common and very, very popular in terms of the molecular biology and the biotechnology studies. And the uh, the very reason why it is because E. coli is easy to cultivate. E. coli is well characterized because this is the first bacteria probably the humans have started working on. So it is having we know a lot of things about the E. coli, right? We know the complete genome sequence we know, right? So it all these things makes it very very well researched microbes. So it is a and that's why it is can be exploited in a multiple way. And then it also has the simple systems which can be manipulated. So genetic manipulation of E. coli is also comparatively easy. For example, you can actually be able to develop the strategies for transformations, transduction, and conjugations, and all that, right? Then talking about the expression system, so considering the E. coli as a host, people have developed the different types of expression system. We have the T7 based system. We have the so we have generated the different types of host molecule. Even we have generated not only E. coli but the different types of host E. coli strains which can be used for the protein production. Now, the fourth is the safety part. So E. coli is very safe because it does not cause any very harmful diseases. It only remain as a harm does not cause any disease actually. So it is not except the pathogenic strains. Some E. coli strains are pathogenic, but majority of these E. coli strains are non-pathogenic. And then the economic considerations. growing the E. coli is very cost effective, and it also gives the very high production of the proteins. And because of that, the E. coli is a very popular host, as been used for the protein production or for propagation of the foreign DNA. Now, talking about the disadvantage, so E. coli is a prokaryotic organism, right? So it does not have the like the the splicing machinery. It does not have the glycosylation machinery and so on. So basically, the protein will not get modified. If you're talking about the the mammalian system and all that, so because of that, you require the eukaryotic system. And when you talk about the eukaryotic system, you go with the simplest eukaryotic system, and the simplest eukaryotic system is yeast. So yeast can also be used as an expression system, right? So yeast is the simplest unicellular eukaryotic cell available for the protein production. It is easy to manipulate, and production cost is also very low in comparison to the other eukaryotic system. So apart from yeast, you also have the insect cell lines. You can actually have the mammalian cells, but these systems are much costlier in terms of the to propagate those cells and all that. And it offers the most of the advantages what are available in the eukaryotic system, but it also offers the advantage in terms of that its production, its propagation is as simple as the E. coli, right? So yeast is very popular in terms of the one of the simplest eukaryotic system. Now, talking about the yeast, you can actually have the two different choices of the yeast. You can have the non-methylotropic yeast, or you can actually have the methylotropic yeast. So, in a non-methylotropic yeast, they don't utilize the one carbon compounds such as methanol. So, it can be utilized to other carbon sources such as glucose, lactose, and maltose, and starch and alkanes. We such as like S. cerevisiae, K. lactis, and Y. lipotica, and these strains are mostly used for fermentation to produce the alcohol. The major advantage of this class is the better understanding of the molecule, because the S. cerevisiae is extensively being studied yeast, and because of that, it can be used for doing the different types of complementation experiments, the molecular biology related experiments, biochemistry related experiments, and the fermentation technologies. But the disadvantage is that since these molecules, these host does not use the methanol as a one carbon source, they cannot used for the protein overexpression, right? So they don't have the protein overexpression ability, right? Non-methylotropic yeast strains. Then we talk about the methylotropic yeast. So the major advantage of this is that they don't utilize one carbon compound such as methanol as a carbon, and these strains have a high level of methanol oxidizing enzyme, and that allow them to go very strong and grow in high density. And the examples are Pichia pectoris and Pichia augusta, and Pichia methanolica and the Trivedi. So this strain can be used for the protein overexpression, right? And so very carefully, these two molecules are being done. So non-methylotropic can be used for the fermentations. Methylotropic can be used for the protein overexpressions. Now, if you see about the yeast as the expression system, so you have the non-methylotropic strains and the methylotropic strains. So in the non-methylotropic strain, you have the S. cerevisiae, K. lactis, and all that. And you also have here also the people have developed the constitutive versus the inducible system. So and they use the different types of promoters. But ideally, the methylotropic strains such as the Pichia pectoris and the Pichia methylica, they use the inducible systems such as OX1 and the MOX promoters for the protein productions. Now, if the some of the problems which are associated with the yeast as the host strain can be solved even before going into the complicated system of the mammalian system, and that is being offered by the insect cell lines. So insect cell lines are actually offering as a expression system. So in the baculovirus expression system, which offers the protein modifications, processing and transport system, which is not actually been offered completely by the yeast, because yeast is a unicellular organism. So its protein modification and processing ability are, or some in some of the cases, they are different from the mammalian system. So compared to the yeast, the downstream processing and recovery of cellular protein is much easier in the baculo system, right? But you require a much complicated procedure for the protein to be produced. For example, in the first step, you are supposed to clone the foreign gene into a transfer vectors. Then you have to generate the recombinant baculovectors. Then you have to screen these baculovirus vectors. Remember that the baculovirus is a virus which infects the insect cell line. Okay, and then it insert the your transfer the vector into the insect cell lines. And so basically it has a advantage also, that once you generate the baculovirus, you don't need to do the transformation. You can just infect the cells with the baculovirus, and it is actually going to transduce your transfer DNA into the cell lines. Then you can do the culturing of the recombinant cell lines, and you can do the protein productions. Then apart from that, you can also have the mammalian expression system. So mammalian expression system offers the advantages of that it is going to be a eukaryotic system, but it has the disadvantage that it does not give you a large quantity of proteins. And on the other hand, it is very, very costly also. So similar to the other expression system, the protein production in mammalian system can be achieved either from a vector present as the extrachromosomal DNA, or the sequence is integrated into the genome through the homologous recombination to establish the permanent cell line. So in the mammalian expression system could be transient expressions or could be permanent expressions. The expression from the transient or the permanent cell line can be from a constitutive or the inducible system. Most of the time, the mammalian expression system is constitutive type rather than the inducible promoters. Irrespective of the expression, in the different steps which require to produce the protein is the cloning of the foreign gene into the mammalian expression vectors, then transfection of a cell line with the recombinant constructs, then screening and selection of the transfected cells, then culturing of the transfected cell, and then the protein production. So remember that the mammalian expression system is mostly being used not for protein production but for studying the basic processes, such as how this protein is produced, the mechanism of the protein production, and what are different components are being involved, and so on. Because the protein production in the mammalian expression system is a very, very costly affair, because you are supposed to grow the mammalian cells, and growing the mammalian cell is costly, and as well as it is also require the lot of infrastructures. You require the CO2 incubators, you require the biosafety cabinets, and you require a dedicated room for doing the cell culture, right? There are different types of cell lines we can use for the protein production, so or the experiments related to the mammalian expression system. So you can have the different types of cell lines. You can have the breast cancer cell lines like MCF7 and so on, right? Mammalian expression system could be a transient expression or it could be a permanent expression. Transient expression means it will go for 72 hours, like since you have done the transfection, or permanent expression where the gene is going to be integrated into the genome, and that's how you can have the permanent expressions. The expression is high in the case of transient expressions, but it is short-lived, right? So the cells transfected by DNA express the protein for 72 hours. Transient expression system is used for screening the cDNA libraries, isolation of a particular cDNA clones, expressing the surface antigen, and to test the applicability of the recombinant construct going to use for the permanent expression. So before you actually go with the permanent expressions, people do the test the particular construct into the transient expression, because transient expressions are easy to do, because you take the mammalian cells, you prepare the recombinant clone, and then you do the transfection and check whether the particular protein is getting expressed or not. Permanent transfections, the permanent expression of a gene is possible if it will be integrated into the chromosomal DNA. The most crucial step to establish a permanent expression system for a gene is to frequency of integration events rather than the number of DNA uptake. So in simpler one, the permanent expression system depends on to the recombinant frequency instead of the transfection efficiency. Recombinant system can also have the constitutive systems or it could be a inducible system. So what we have discussed so far, we have discussed about the different types of host system, right? We have discussed about the E. coli as an expression system. So we discussed about the host, what are available for the protein production once you have generated a recombinant clone, right? So once you generated a recombinant clone, you can use the E. coli as the expression system, you can use the yeast as the expression system, you can use the insect cell line as the expression system, you can use the mammalian expression system, and number 5, you can also use the plant as the expression system. But majority of the time, the plants are never being used for as an expression system. Plants are always being used for expressing a particular protein so that it helps the plants to change their phenotype, right? Such as like you express some protein and then it becomes resistance for the abiotic stress, or it actually ease out the abiotic stress, or it actually take care of the deficiency of some of the metabolites and so on, right? So plant is not very common in terms of utilizing as the expression system compared to the other expression systems such as E. coli, yeast, or insects, or mammalian cells. So in the two component system, so far what we have discussed about the host cells. Now we'll discuss about the transforming agents, because that is also very, very important. You have to understand what is there in the transforming agents. So transforming agents are going to be also different types. You can have the prokaryotic transforming agents, or you can actually have the eukaryotic transforming agents. So vectors, the vector is the which is actually also being called as transforming agents, has two responsibility. One, it is actually being used for ability to carry the foreign DNA, and number two is ability to replicate into the host. Hence, to fulfill these responsibility, a number of properties are desired. So what is the criteria, or what is the properties you should have so that you can be able to use these vectors into the two component system? So the criteria for the good vector. So number 1 is the low molecular weight. The low molecular weight or size confers a number of advantages. Small size vector is robust towards the shear stress and easy to handle. In addition, after the ligating foreign DNA into the vector, the size of the resulting recombinant DNA will be small and it will be easier to deliver the recombinant DNA into the host. So remember that the transformation efficiency, right? Anyway, we discuss about the transformation, but transformation efficiency is directly proportional to the size of the DNA. Okay, so if the size of DNA is big, the transformation efficiency will go down. So what is mean by the transformation efficiency is now how many molecules of DNA will enter into the bacteria, right? Then number 2 is post entry into the host should give the phenotypic change. So another important feature is that the vector DNA should give additional phenotypic changes in the host cells so that the recognition of the transformed cell could be easier. So selection is also easy, should be easy when you're talking about the vector. So when you are designing a vector, right? First is it should be of low molecular weight. The second is once it enters into the host, it should give you a phenotypic changes so that you can be able to say visually that okay, this is transformed cells, this is a untransformed cells. Then number 3, it should be have a multiple cloning sites. So it should actually have a stretch of DNA which contains the restriction site for possible site of insertion of the foreign DNA. So more number of multiple cloning site, more it gives a more flexibility in terms of using that for the integration of the foreign DNA. And then number 4 is it should be a high copy number, right? So a high copy number is required because then it will give you the more amount of DNA, and as well as so that you can actually be able to use this for the production of more amount of DNA. Now, if this is the criteria, you are supposed to have the multiple components which you are going to put into a vector, and then only it is going to use for the for taking for the mammalian clonings. So what are the components required for a vector? Okay, so the first is you are requiring the origin of replications, right? Remember that the origin, the DNA is going to be replicated in a circular DNA, right? Has to be replicated from a particular point, right? And this point is called as the origin of replication, such as ORIC, right? So ORIC is a origin of replication into the bacterial system. So if you have a if you have a linear DNA, right? Even if you have a linear DNA, the DNA is getting melted by the helicases by some place, okay? And this place is being called as the original replication. From this point, one replication fork will go in this direction, the other replication fork is going in this direction. Similarly, in the bacterial system, it will actually going to be made the single standard, right? And then the one replication fork will go in this direction, the other replication will go in this direction. So this original replication is very important. So like any other replicating DNA, the plasmid DNA needs its own independent original replication to provide the replication start to make the more copies. It decides the range of bacterial host strain can be used for particular. So this original replication only decides the number of hosts it is going to be comfortable or it is going to be used, right? So for example, the plasmid containing the origin of replication from the col E1, this is a bacteria, can be able to grow in limited number of bacterial species such as E. coli, right? In contrast, the plasmid containing the original replication from the RP4 or RSF1010 can be able to grow in most of the gram-negative as well as the gram-positive bacteria. So the diversity of the original replication is going to decide in how many different types of bacterial species or the other host strains this particular vector is going to replicate. Then the number 2 is the selection markers, right? So selection marker is the form of either antibiotic resistance gene or the enzymatic DNA is being the phenotypic changes in the host after the entry of the plasmid. Then the number 3 is the promoter. So suppose you are use this vector for the protein production, then you also require the protein production related machinery, right? So then you require the promoter also. The plasmid replication in the host is performed by the host provided proteins such as DNA guidase, DNA helicase, polymerase, and DNA ligase. But protein required for conferring the antibiotic resistance or enzyme used for selection is on plasmid, and a promoter adjacent is required to express the gene present on the plasmid DNA, right? Promoter is also needed to express the gene present on the foreign DNA. Now, keeping this into account, right, you can have the different types of vectors, right? Remember, in a two component system, you actually require the vectors as well as the host, compatible to each other. So as vector need to be replicated in different host strains, the vector needs a special additional structural feature to make it suitable for a particular. So the question is why one vector does not replicate in different host strains? Because it does not have the origin of replication. So it requires the origin of replications compatible with the hosts, right? So the replication of the host vector is controlled by the original region and it needs to be recognized by the host factor, especially the DNA polymerase, to perform the replication. So consequently, there are different types of vector DNA to suit the cloning of a foreign DNA into a particular host strain. So you can have the bacterial plasmids, you can have the phage-based vectors, you can have the yeast vectors, and you can have the mammalian vectors. Okay, so this is the first, the plasmid which is being synthesized by the humans, right? This is the first plasmid being synthesized. Giving you the examples or the link also for the, for the paper where the construction of this particular plasmid is being described, and it's very interesting and important to read how the humans have synthesized this first plasmid, which is called as PBR322, by deriving the, the molecules or the DNA component from the three different, the bacteria. So it has taken the PSC 101 derived material. This is the PSC 101 derived material. Then they have taken the RSF 2124 derived material, and this is being called as PMB derived material. So they have taken, so there are plasmids which are present in the bacteria, right? So they have taken the individual component from these two things, and then they have developed the PBR322. And so PBR322 is a 4,359 base pair long plasmid, and it has a 40 unique recession sites. 11 recession sites are present within the tetracycline resistance gene, and the 6 sites are present within the ampicillin resistance genes, right? So basically, it has the two restriction sites, two antibiotic resistance genes. One is the ampicillin resistance gene, the other one is the tetracycline resistance genes, and then it also has the origin of applications, right? And in addition, the two sites are present within the promoter of the tetracycline resistance genes. Cloning any DNA fragment to disrupt the resistance gene, and as a result, it can be used as a criteria for selecting the recombinant plasmids. So, what is the application of the PBR22? Remember that the PBR22 does not have the promoter, so it does not have a promoter, and as a result of that, it can only be used for the, the cloning purposes, right? And it is used to study the transcription and translation of the prokaryotic genes, and it is the primary source of design and construct improved plasmids for the specific applications. So from the PBR222, people have developed the PUC19, and so PUC19 is the initial examples of the bacterial plasmid, which is size of 2.8 kb, and it has the multiple cloning site. Then it has the LAG-Z, which actually allows him to go with the blue-white screening, and then it also has the multiple cloning site and so on. And PUC19 vector also has a small stretch of DNA which encodes for the rapid detection of insert by the blue-white screening. So this is a LAG-Z, which is going to be used for the blue-white screening. Okay, and then we talk about the eukaryotic vectors. So you can have the yeast vectors, you can have the phage-based vectors, and we can also have the mammalian vectors. So in the eukaryotic system, you have the two possibilities. One, the vector could be present as an extra chromosomal DNA, or the vector which is going to be the integration vectors. So vector as an extra chromosomal DNA, these vectors remains in the erotic cell as extra chromosomal DNA and express the protein. So these vectors are actually be present as the transient in the transient expression system. Okay, whereas these ones are probably be present in the permanent system, where you are actually putting them into the integration vector. So these vectors carry the integration site to facilitate the recombination mediated integration into the chromosomal DNA of the host cells. So one of the eukaryotic plasmid is yeast, right? So yeast vector system, these are having a couple of similar features such as presence of multiple cloning site. You can have the shuttle vectors. Shuttle vector means the vector which actually has the origin of replication for the E. coli and the yeast, which means this particular vector, these vectors can be able to grow into the E. coli, and they can also be able to grow into the yeast. And then you also have the selectable markers. So there are three different types of yeast vectors. You can have the episomal vectors, you can have the integrating vectors, and you can also have the yeast artificial chromosomes. So in the episomal vectors, yeast episomal vectors are constructed by combining the bacterial plasmids either with the yeast 2 micron origin of replication or with the autosomal replicating sequences. So this is the YEP24, which is actually a yeast episomal vector. And to 2 micron based episomal vector. It is a 6.3 kb plasmid with a copy number in the range of 5200. And these plasmids are much more stable than the ARS based or the autonomous replicating sequences based plasmids. Then we have the integrating vectors. So if episomal yeast vectors are present as an extrachromosomal DNA and are unstable, this can overcome by integration of the vector into the host chromosome. So in the yeast, integrating occurs by the homologous recombinations. In yeast, integrating plasmid contains the target sequence for the integration into the chromosomal DNA, selectable marker, and the bacterial origin of replications. Before vector delivered into the yeast, it is digested with a unique decision endonuclease to produce a linear DNA to increase the transformation efficiency and the integrations. And then we also have the yeast artificial chromosomes. So yeast artificial chromosome or the YAC is a vector of choice used for the to clone very large DNA fragment. Remember that the YAC is being used for the integrating the genomic preparation of genomic DNA, right? Especially from the plant or the other organisms where you have the large genomic DNA, right? YAC vector is like a chromosome. It has the ARS sequences and centromere sequences and telomeres at the two ends to give the stability. It has an ampicillin resistance genes for selection into the E. coli, and it also has the E. coli original replication for propagating in bacteria. In addition, it has the autonomous replicating sequences sent for the centromere functions and the URA3 and TRIP1 for the selection into the yeast. So it's actually going to use this for the oxotrophic selections. For cloning, the YAC is digested with the ESMA1 and the BAMH1 and the alkaline phosphatase to generate a linear plasmid. Now, foreign DNA is added for the ligation, and the recombinant DNA will grow will allow to grow on the URA3 minus or tryptophan minus to grow on a uracil and tryptophan deficient media. So basically in a YAC vector, what you have is you digest this with the ESMA1 and BAMH1, it is actually going to form the linear DNA, and then you integrate your input DNA, and that input DNA is going to integrate into these sites, right? And then you can actually, and because of this is going to be present, it is actually going to be able to synthesize its own uracil and tryptophan, and as a result of this will be able to grow onto the uracil and tryptophan deficient the media, whereas this will not. Okay, so, and as a result of this you can be able to screen them. Then talking about the another eukaryotic plasmid, which has actually been called as baculovirus system. So in the baculovirus system, the baculovirus is a rod-shaped bacteria, a rod-shaped virus, which infects the invertebrates, including the insect cells, right? So this is the baculovirus system where you have the the cloning site, right? So in the center you have the cloning site, and then you also have the promoters, and you also have the termination sites, and this whole block is being used. So this is the the part of the vector, part of the virus, and you can actually integrate your foreign DNA into this particular site. So this can be replaced with this, right? So post-infection, the virus is either released as a free viron, or many virus particles are trapped in a protein complexes such as polyhedron, right? The protein responsible for trapping the virus into polyhedron is polyhydrin, and it helps in the transmission of viruses from the one host to other. The polyhydrin is not important for the virus propagation, but it is very under a strong promoter to produce the protein in a large quantity, right? So because this polyhydrin protein, which is actually present inside the virus, is present along with the strong promoter, right? But this polyhydrin protein is useless for the virus. So what they have done is they have replaced this with a multiple cloning site, and then you can actually be able to put your DNA here, right? So if you put your DNA of interest, instead of producing the polyhydrin, they will start producing the protein which is corresponding to this particular DNA. And So the baculovirus, the Autographa california, multiple nuclear protein or ACMP is used as a vector to express the protein. The transfer vector map is given, right? This is the vector map. The gene of interest will be inserted into the cloning site placed adjacent to the promoter, and it has a polyhedron termination sequences downstream to the cloning site to stop the transcription of the clone gene. And so what we are going to do is, so this is the, the mammalian vectors, right? So in the mammalian vector, the large number of excellent mammalian vectors are in circulation to the eukaryotic gene for protein synthesis and study of the transcription mechanisms. It contains a eukaryotic replication of origin from an animal virus such as SV40, a promoter to derive the expression of foreign gene, and a selection marker, other eukaryotic features such as polyed dilations, transcription termination, etc. site, right? So you actually have a multiple cloning site, you have the promoters, you have the, and then you also have the selection marker like the ampicillin, and you also have the original replication for the eukaryotic system, you also have the original replication for the E. coli system. And then we have the bacteriophage-based vector. So in the bacteriophage-based vector, you basically are replacing this non-essential region, right? This non-essential region with the with the the gene of your interest, right? So bacteriophage genome. So bacteriophage is a linear double-stranded DNA of 48.5 kb. On both end of the genome, it has a stretch of 12 nucleotides which are complementary to each other, right? So basically you have the left cohesive ends and you have the right cohesive ends, and with the help of these, basically they can actually be able to circularize. So these sites are being called as COS sites, and it allows the circularization of the viral genome after entering into the host cells. Genes are arranged in between these two cohesive ends and codes for the protein responsible for making the head, tail, factors for recombinations, and the process of lysosine. The central region of the genome is non-essential and can be replaced without much affecting the growth and the infectivity of the viruses. So basically this small portion, you basically have the small region which can be replaced. So as a result of this, this region can be exploited to develop the cloning vector, right? And so these are the two different types of vectors. You have the EMBL3 vectors, and you also have the EMBL4 vectors. So basically you have the left arm, you have the right arms, and then the central portion which you can use for inserting the inserting the foreign DNA. So what we have discussed about the transforming agents, we have discussed about the host, right? And but transforming agents and the host and the production of the recombinant DNA requires different types of enzymes, right? You basically require the different types of molecular biology enzymes. Like, for example, you require the polymerases, you require the restriction enzymes, you require the ligase, right? You are using this here, right? And then you also require many more enzymes, which you require for production of the recombinant DNA. So what are different types of enzymes you require? You require the restriction enzymes for cutting the DNA at a specific site. You require the polymerases for doing the PCR amplifications. You require the alkaline phosphatase for the removal of terminal phosphate groups. And then you also require the DNA ligase for joining of the two DNA strands. Now, restriction enzymes are actually be a part of the prokaryotic host defense mechanism, right? And that has been exploited for the molecular cloning. So basically, every bacteria or every prokaryotic system has the restriction methylase system, which actually allows him to distinguish between the self and the foreign DNA. Although the precise mechanism is not known, but in the absence of methylation, a closed complex is formed and allow the proper activation of the cleavage. But presence of methyl group, whether it is the hemimethylation or the non-methylated, the nucleotide does not allow the formation of closed complex, and consequently the enzyme fall into from the DNA. So this is what exactly happened, right? So if suppose this is the restriction site, right? So when the restriction enzyme comes, it actually checks whether the adenine present within this site is methylated or non-methylated. So for example, if this is the DNA, right? In this one, this DNA is actually non-methylated. So if this is the unmethylated DNA, right? If it's the unmethylated DNA, the enzyme will go and see and then recognize, and then it is actually going to cut, and then it is actually going to produce the two fragments. One is which is complementary to each other, and they will actually going to have the the coincidence, right? So this is the fragment what it's going to generate, right? Similarly, this one also going to have this fragment. Similarly, if you have the methylated DNA, right? For example, if you have the methylated DNA, whether it is fully methylated or hemimethylated, then the restriction enzyme will go and bind, but will not be able to form the close complex. And if you cannot form the close complex, then it will not be able to cut. And this DNA is going to be called as self, whereas this will be called as foreign DNA. So this is a kind of a defense mechanism what is present inside the bacterial system. And this is what the mechanism is saying, that how the mechanism, so it's actually going to recognize the cleavage site, then it is actually going to bind to the respiration site, and then it is actually going to catalyze the cleavage site. Only when the when the adenine is not methylated. This I think I will skip. Okay. And what is the property of the restriction enzymes? So restriction enzymes are recognizing the sequences which are palindromic in nature, right? So palindromic, what is mean by palindromic is that if you read them in the forward direction or if you read them in the reverse direction, they are going to be same. So example, this is the restriction site, right? So GGA-TCC. So if you read from this side, right, it is going to be GGA. Similarly, if you read from the reverse side, right, then it is going to be called as GGA. So these are actually being called as the palindromic sequences. And so, for example, the BAMH one has a restriction site as GGA-TCC, and on a strand 1 and a strand 2 will have the read the same sequence. So if you read from this direction or if you read from this direction, it is going to be same. So GGA-TCC, similarly GGA-TCC. So this kind of phenomena is called as palindromic sequence, and it has a lot of applications. Because of this only, you can be able to generate the sticky ends. So for example, if it cuts here, right, it is actually going to be generate the sequence like this. So DNA is going to be like this, right? Other DNA is going to be like this, right? So because of that, it is actually generating a sticky ends, right? And that's how it is actually going to, allow the religations between the foreign DNA and the other DNA. And then the second is sticky ends, right? So the type restrictions like cut the DNA together to generate the DNA with a hanging DNA stretch with 4 to 6 nucleotides. And these stretches contain the fragments are cohesive to each other. For example, when you have GGA-TCC and when you cut the fragment with BAMH1, so you will cut here and you will cut here, right? And as a result of that, you are actually going to have the strand like this. So this is the overhang what you have. And then similarly, the other one, you are going to have this. So imagine that if this belongs to the vector and if this belongs to your DNA, the foreign DNA, and if you put them together, right, they will come and type. So this G is going to recognize this C, this A is going to recognize this T, this going to, right. Similarly, so they will come together, and that's how they are actually going to form spontaneously they will form the double stranded DNA, and as a result, they will replicate inside the cell, right? How you're going to set up the restriction reactions? So restriction reactions require the DNA which you want to digest, then you require the restriction enzymes, then you require the buffers, then you require the BSA, and then you also require the sterile waters and so on, right? And restriction reactions are always being done in a large volume such as 50 microliters. So you put like DNA as 1 microgram, you require the 0.5 to 10 units of the restriction enzymes, whether you are doing the one enzyme or the two enzymes. And then you also require the buffer, which is 1x. So normally the buffer what you get from the companies are 10x, right? And then you also require the BSA, which is supplied as 100x. So you require the 1x. And then you require the sterile water to make up the volumes, right? And in this one also, you are actually going to set up the reaction in a particular sequence. So for example, you are going to take the water first, right? Then you are going to add the buffer. Then you are going to add the restriction enzymes. Then you are going to add the BSA. And then you are, at the end, you are going to add the DNA. And then you will incubate this at 37 degrees Celsius for whatever the desired time. And then it is actually going to give you the digestions. Now, what is mean by the star activity? So some of the restriction enzymes are actually, for example, if you have a restriction enzyme which is going to cut as GGATTC, right? Sorry, TCC, right? So it has supposed to cut after G, right? And the same is for the complementary sequence, right? So it's supposed to cut at after G. But in some of the conditions, when it is not recognizing the G, it is actually going to cut here, it's going to cut here. So it is actually not going to cut as per its documented restriction sites. In those kind of aberrant activity is being called as star activity. It happens because the restriction enzymes are being supplied in the 50% glycerol. So if the glycerol value is more than 10% in the reaction, then it is actually going to form the star activities. Then the second enzyme, which is very also important for the molecular cloning, is the ligase. So ligase is joining the two DNA to generate the chimeric DNA in the basis of the cloning. It is essential step to generate the clone containing the foreign DNA. When cohesive and generated by the action of restriction endonuclease associated with a nick is going to be sealed and give the complete circular DNA, right? So what you see here is that when you are actually generating this, right? Remember, now, if you are cutting with the BAMH one, right? So what you are going to generate, right? You are going to generate G, then C, C, T, A, G, right? So this is when it is going to fit with another one, right? It is going to fit with another one, right? G, A, T, sorry, G, then C, like that, okay? So when it is actually going to sit with the other one, right? There will be a gap here, right? So there will be a gap between these two. There will be a gap in between these two, right? This is a gap which is going to be filled by the ligase, right? So what ligase is going to do is it is actually going to bring these two molecules together, and that's how the phosphodiester linkage is going to be formed between these two molecules and these two molecules. So what it is going to do is it is going to take up the ATP, right? It is going to bring these gaps, right? So on one side you are going to have the OH, the other side you are going to have the phosphate, right? What it is doing is it is just bringing them together so that there will be a phosphodiester linkage what is going to be formed between these two molecules, and that's how it is going to be a complete double stranded DNA. So you can actually have the two different types of ligase. You can have T4 DNA ligase or you can have the E. coli DNA ligase. Both of the ligase are doing the similar function except that they are using the different types of cofactors. In the E. coli ligase, it is using the NAD+, whereas T4 DNA ligase, it is using the ATP. How you are going to set up the ligase reactions? So ligase reactions are completely opposite to the restriction enzyme reactions, right? So remember that the restriction enzyme reactions, we are doing it in 50 microliter, but the ligase reactions, we are doing it in the small volume because we want that the fragments should interact with each other in more, a crowded state. So you basically want to increase the molecular crowding, and because of that, you actually going to set up the ligase reaction in a small volumes. So you're going to take the vector, right? So this is the vector. You're going to take the vector, you're going to take the insert, and ideally we always take the vector versus insert in a range of 1:3, right? In terms of moles, not in terms of molecule, the weight, or in terms of quantity, okay? So that you have to calculate. So vector is size whatever, is insert size whatever, then you have to take into 1:3 ratio. Then you have to take the enzyme 0.5 to 10 units. You have to take the ligase buffer, which actually contains the ATP as the cofactor, right? And then you also require the BSA, and you require the sterile water and ligase, right? And how they are going to set up the reactions? So you are actually going to take the water, you are going to take the buffer, right? So number one, you're going to take the water. Number two, going to take the ligase. Then you are going to take the BSA. You're going to take the enzyme. You are going to take the vector and insert, right? And some cases, what we do is we actually heat up the reactions so that the DNA is going to form the single standard DNA, and then we cool down, and that actually promotes the ligation reactions. Apart from this, we also require some other kind of enzymes which is required only for doing the molecular biology manipulations. One such enzyme is alkaline phosphatase. So remember that the nucleotides are actually going to have either the OH, which is on the 3' end, or you can actually have the phosphate, which is on the 5' end, right? In some cases, when you are doing the digestion with a single enzyme, you are actually going to generate on the two fragments, one fragment which is going to have the phosphate, the other fragment which is going to have the 3' OH. So if you don't do anything, right, they will be having the spontaneous affinity for each other, right? For example, if I have a vector and this I have digested, right? So if I have a vector which I have digested with only single enzymes, right? So for example, if I have digested this, so this is the double stranded DNA, right? If I have digested this only with the E. coli, right? If I have digested with the two enzymes, then it is not a problem. But if I have digested only with the E. coli, then what will happen? It is actually going to have the overhang, right? And it is actually going to have the E. coli side on this side. It is going to have the E. coli side on this side, right? Now, on this side, you are going to have the OH. On this side, you are going to have a phosphate. Similarly, on this side, you are going to have a phosphate. On this side, you are going to have a OH. Now, even if you don't do any ligation reactions, this molecule has a strong affinity for this one, and this one is also having very strong affinity for this one. So basically, even if the insert is not present, it is actually going to get recircularized, and it will give you the background in terms of the false positive, because ideally it should recircularize only when you are putting the insert, right? So if you are putting the insert with also digested with E. coli, then only it should form the the double standard DNA, right? Then only it should form the double standard DNA. But if you have that same enzyme on both the sides, then even without that also it is going to recircularize. To avoid this, what we are doing is we are removing this phosphate from the vector, okay, from the plasmid, okay. And this is be the function of the alkaline phosphatase. So alkaline phosphatase is an important enzyme. It's has a lot of function, but its main function is to remove the phosphate, okay. And this is the reaction conditions here where you're going to use this, right? And the alkaline phosphatase is always being used for avoiding the recircularization when you are doing the single restriction digestion. Then we have the polynucleotide kinase or PNKs, right? So remember that when we were discussing about the probe formation, right? We have not discussed one of the reaction, which is the terminal transferase, right? So similarly, we have the kinase also. So this is very important for the different types of applications. But in the molecular biology, is being used for attaching a phosphate group, right? So PNK is doing exactly opposite what alkaline phosphatase is doing, right? So in some cases, you have a 5' OH, but that 5' OH will not ligate because you require a 5' OH. So it is actually having the reverse effect of the alkaline phosphatase, adding the phosphate group onto the 5' prime. So magnesium acts as a cofactor for the enzyme, and it is crucial for its activity. And this is the reaction condition for the polynucleotide kinase, where you require the DNA, then you require the buffers, you require the polynucleotide kinase, and you require the ATP. And the same thing can be done even for labeling the DNA with the phosphate group, because you can take the radioactive ATP and it will actually going to label the DNA with the radioactivity. Then we also have the terminal transferase. So terminal transferase is also doing the the adding the terminal amino acids. It's an enzyme which plays a crucial role in the immune system and so on. It's a DNA polymerase which adds the nucleotides onto the 3' of the DNA without a template. So this is a unique enzyme which actually has no template required, right? And because of this only, the terminal transferase is being used for preparing the the cloning reactions. So enzyme can add a similar nucleotide to form a homopolymer tail on the prime end, right? So unlike most nucleotide, it does not require the template. So this is the reactions. You require the DNA, you require the reaction buffers, you require the enzyme, the NTPs, and nucleus. And you can actually be able to use this for generating the vector for the TA cloning. And TA cloning is a very, very important, tool for, facilitating or simplifying the reaction mixtures for cloning actually. And then we also have the poly A polymerase. So that actually is same as terminal transferase, but except that it only going to add the adenine, not the other molecules. So this is all about the molecular cloning. We have discussed so far is, um, the what we have discussed. We have discussed about the two component system. We discussed about the vectors. We discussed about the host, and with this, I think you can understood that how you can be able to do the molecular, cloning, and you can be able to generate the clone. In the second part, we will discuss more about how you can be able to transform, how you can deliver the DNA, how you can use that for protein production. But before that, if you have any questions, I'll be happy to answer.
Now, sir, if anyone wants to ask any question, please, y'all can ask before sir proceeds. Okay, still no questions have come up. So, sir, I have one question. Sir, can you please explain, in one of your slides, you have mentioned blue-white screening. So, can you please tell what is blue-white screening?
I think we will take up that in detail when we'll talk about this third part. So we will discuss in blue-white screening in detail.
Thank
So
you,
that
sir.
time I think you will be able to understand.
Okay, sir. Thank you, sir. And still no questions have come up. So, sir, I can think you can move to the third
Yes,
part.
yes. Okay. Yeah.
Thank you, sir.
Okay. So without wasting time, because so in the third part we will discuss about the overexpression. Okay. So far what we have done, we have actually produced the clone, right? So we produce the clone, right? We discussed about the enzymes, what you require for the molecular cloning. We discuss about the two component system. So we discuss about the transforming agents. We discuss about the host. Now what we'll talk about is we'll discuss about the process, right? How you, once you got the recombinant clone, how you are going to process this for the subsequent steps. So first step is the DNA delivery into the host, right? So we started from here, right? In the morning, right? We end up here, number 2, right? So we discuss about all this cloning stuff, right? Now we'll enter into this where we'll discuss about the DNA delivery and screening, and then subsequently we'll discuss about the overexpressions. Now talking about the DNA delivery, right? When you want to do a DNA delivery, you have to actually work on
Excuse
the,
me, sir.
yes.
Sir, we can't see your slides. I think you
Oh,
haven't shared the screen. Okay.
okay. Thank you. Thank you. Thank you. Is it fine now?
Yes,
Yes,
sir.
sir.
Yes, sir.
Yes,
Okay.
yes.
Okay, so this is what we were saying, that from the morning we were discussing about the molecular cloning, right? And we were discussing about how to identify the gene. Then we discussed about the cloning part, right? Then we discussed about the DNA delivery, right? So in now we'll just going to start with the DNA delivery part, and then we'll discuss about the overexpression part. So when you talk about the DNA delivery, you have to work on two things. One, so see, you have to insert this inside this, right? So, and if you want to insert this into inside this, you have to talk about the chemistry of this plasma membrane, chemistry of this barrier, right? Because if you overcome the barrier, the DNA will enter, right? So basically the surface chemistry of the host cells, the host cell surface chemistry either will attract or repel the DNA as a result of opposite or the similar charges. So imagine that if the host the plasma membrane is negatively charged, it is going to repel the DNA. If it is positively charged, it is going to attract the DNA, right? So in the presence of cell wall, it causes the additional physical barrier to take up an entry of the DNA, and that's why you require to prepare a competent cell so that the host cells should be able to take up this DNA. Similarly, you have the charges on the DNA, which is negatively charged, right? So DNA has a negative charge because of the phosphate, and this negative charge also has to be neutralized. And this is the mechanism of transformations. So you actually have the different types of reagents which you can use for preparing the competent cells. So you can actually use the for different types of bacterial stain. For example, you can use the Streptococcus pneumoniae, you can use the mitomycin C and fluoroquinolone. Then for the Bacillus subtilis, you can use the UV light, you can use the Helicobacter pylori, you can use the ciprofloxacin. For Legionella pneumonia, you can use so many of these molecules. And then for the E. coli, you can use the calcium chloride or the rubidium chloride. And all these you have to do for that you can actually be able to take care of the surface chemistry of the host cells so that they will be happily take up the DNA. How you're going to prepare the competent cells? So bacteria is incubated with the divalent cations such as calcium chloride or magnesium chloride or the rubidium chloride for 30 minutes at 4 degrees Celsius. And remember that the bacteria is following a growth curve, right? Where you have the initial lag phase, then you have a log phase, and then you have a stationary phase, and then you have a death phase, right? Uh, this is the only phase which you are going to use because here the bacteria is very active and it is actually dividing. So the cell wall of the treated bacteria is swelled. So when you do this, incubations with the divalent cations, the bacteria is going to swell and it is going to gather the factor which is required for the intake of DNA, which is docked onto the plasma membrane. Because you are doing all these reactions and because of that the bacteria is going to swell and its cell wall is also going to be compromised. It is very fragile, so they are very fragile. If you do little bit any kind of shaking, they will actually going to die. Then the storage also has to be done at the 15 to 20% glycerol, and you have to store it at minus 80 degrees Celsius. And because they are very fragile, you have to do the centrifugation at a very slow speed, and then the pipetting also has to be done with a cut pipette. So remember that every pipette has a tip, right? And these tips are very pointed at the end. So when you are dealing with the competent cells, then these tips has to be cut in the front so that the bore what you are going to use, so basically you have to cut from here so that the bore what you are going to use is slightly not that thin, so that the the coli cells should experience any kind of shear stress, and if they do, then it is actually going to lyse the cells because they are already fragile in terms of their osmomolarity, because their sensitivity for the osmomolarity is not there because of cell wall, but when you are treating them with the divalent cation, the cell wall is also compromised. And this is what I have shown already, right? So transformation is on. So once you prepare the materials, competent cells, then you are going to do the transformations. So you take out the competent cells, incubate it with the DNA, right? and then you incubate it on on ice for 30 minutes. Then the second step, then you are going to do the heat shock, right? So then you are going to do a heat shock, 42 degrees Celsius for 90 seconds. And when you do the heat shock, the plasma membrane is going to, expand, like, because, heat shock, when you do, the lipids are going to be, a little far away. And because of that, the DNA is going to enter inside the cell. Okay, I think somebody is asking how long we can store the competent cells on -80. You can actually store the competent cells on -80 for an indefinite period of time, uh, as long as you can be able to maintain the temperature of -80. Okay, so then you basically have to do the heat shock, and that actually is going to induce the upregulation of the factor which is responsible for the DNA recombination and the repair. Then in the second step, once the heat shock is over, then you are going to add the chilled media. So that is actually going to induce the recovery of transformed cells, and then you are going to plate these cells on a solid media containing the selection markers. So in this case, for example, you can use the ampicillin, and then you allow these to grow for 18 to 24 hours. Because you have added the selection marker, only the recombinant DNA containing cells are going to grow. The normal host, like the normal E. coli, will not grow because it does not have the resistance. And ultimately, you're going to get the colonies. And once you got done the transformation, so this is the untransformed cells, this is the transformed cells, right? And you will get the colonies. So you see this white color thing, right? These are the colonies, right? Now what you have to do is you have to count these colonies. So it could possible that in some cases the number of these colonies could be very high. In those cases, what we do is we actually divide this into 4 or 8 sectors. Okay, and then what we do is we count the colonies in this sector, we count the colonies in alternate sectors. Okay, and at the end we are going to count and then take the average. Okay, and that is you are going to get the total number of colonies. Okay, total number of colonies. Now you know how much DNA you have transformed, right? So that is going to use for calculating the transformation efficiency. The number of colony forming unit obtained by transforming the 1 microgram of plasmid into a given volume of competent cell is being called as transformation efficiency. For example, if you transformed 1 microliter of 0.01 nanogram per microliter plasmid into 100 microliter of competent cells, you added 900 microliter of LB. At the transformation procedure, we are adding the chilled LB, right? And get a total reaction volume of 1000 microliter, and then plate the 100 microliter of these transformations, right? The plate has 450 colonies, right? That means you got the 450 colonies from the 100 microliter which you plated from 1000 microliter, right? So the nanograms of DNA, what you plated is 1 microliter into 0.01 into 100 microliter out of 1000 microliter. And if you see this, you have actually calculated the 0.0001 nanogram DNA, right? So from the 0.001 nanogram DNA, you got the 450 colonies. So from the 1 microgram, how much reaction? So that you calculate. So then the transformation efficiency is 4.5 into 10 to the power 8 colonies forming unit per microgram of DNA. So this is being called as transformation efficiency, which means if I transform the 1 microgram of DNA, I will get these many number of colonies. And that transformation efficiency is a criteria to decide which one is a good, whether the competent cells are good or bad. Then you can also do the electroporations, right? So you can actually, pass the electric field, and that also is going to do the electroporation. Then we also have the transformation methods in other strains such as yeast. We also have the, and then we also have the mammalian cells. So in the mammalian cells, you are always doing the transfection rather than the transformations. And of transfection, you can have the chemical transfection method, you can have the liposome or the lipoplex different method, you can have the bactofactins, or you can actually have the transductions. I will skip actually this so that, okay. So once the data delivery is over, right, you are actually going to get the host, right? And then you are actually going to put the selection pressure so that you can be able to screen only the positive clones. And then you can actually be use that for overexpression. How you're going to do the screening? So screening of a recombinant clones are utilizing the three important criteria. One, they actually can go with the enzyme, so that enzyme is going to give them a readouts. The second is they can actually have the antibiotic resistance genes, and that antibiotic resistance genes will allow only the host to grow if it is containing the particular recombinant DNA with containing the antibiotic resistance genes, or it actually can give you the phenotypic changes so that it can also be able to give you the different types of colors. Such example is blue-white screening. So in a blue-white screening, you are actually going to do the phenotypic changes. So blue-white screening is always using a chromogenic substrates. So use of the chromogenic substrate to detect a particular enzymatic activity is the basis of the screen desired clone. The most popular system to exploit this feature is the blue-white screening, whereas a colorless substrate is processed to a colored product. So blue-white skinning is being done because there is a substrate which get converted into blue, right? So when you add the X-gal, it get processed into a blue stain, right? So the colorless compound, which is called X-gal, or chemically it is being called as 5-bromo-4-chloro-3 endolyl beta-galactosidase. So it is a substrate for the by an enzyme which is called as beta galactosidase. So when the beta galactosidase process this particular substrate, it produces a big blue color compound, and that these blue color compound get trapped inside the bacteria. So if the beta gal or beta galactosidase is intact, it will give you the blue colony. If the beta galactosidase is being disrupted, then it is actually going to give you the white colony or colorless colony. So beta galactosidase is the product of laxi gene of the lac operon, and it is a tetrameric protein, and and it has an initial N-terminal region which is called as alpha factor or alpha peptide. Okay, but this initial portion is being removed, and then you are cloning that initial portion into a different vectors so that you can complement the defect to form the active enzyme. So if the vector containing peptide will be transformed into the host containing the remaining laxi, the two fragments will constitute to form the active enzyme. This means when they will form the active enzyme, you are going to get a blue colony, right? And when you insert a gene, you are actually going to destroy the alpha peptide, and as a result of that, the complementation will not be possible, and as a result of that, it is actually going to give you the white colony. So this is what exactly happened, right? So in a normal plasmid, what you have is you have the uh, you have the uh, the beta galactosidase, right? And this portion, which is this portion, right, is being deleted, right? And this portion is being placed onto a onto a vector, right? So when these two components come together, they will actually form the active beta galactosidase, and as a result of that, it is actually going to process the X-gal to a blue color product, and this blue color product is going to accumulate inside the E. coli, and it will give you a blue colony. Now, if you insert a host here, if you insert a fragment here, right, then what will happen? You are actually going to have a fragment here, which is going to be inserted within the beta galactosidase, right? So in that case, even if the active beta galactosidase is present, it is not going to be get active because you have already inserted a vector here, inserted a fragment here, and as a result of that, it will not be able to convert the X-gal, and that's how X-gal is going to give you the colorless product. Okay, which means X-gal will remain unprocessed, and because of that, it will give you the white colony. Okay, so if the X-gal is active, it will give you blue colony. If X-gal is inactive, it is going to give you the white colony. And then we also have the antibiotic sensitivity. So you can have the different types of antibiotic genes, and that gives you the sensitivity against the different types of antibiotics. Then we also have the insertional inactivation that anyway we have discussed just now with the LAG-Z, right? And then we also have the antibiotic resistance genes. Then we have the insertional inactivation of CI. So this is another repressors which are present in the viruses, and that also can be used for selection. Then we have the complementation. So in the complementation, what you're doing is you are actually, uh, doing like you are, if the enzyme is active, it is actually going to process the inhibitor to a toxic product. But if the enzyme is inactive, it is actually going to not be able to convert the inhibitor into a toxic product, and as a result that the cells are going to perform. And then we also can use the screening of transfected mammalian cells. So you can actually use the reporter gene assays, or we can also use the GFP. So, and we can also use a luciferase based system. And this is the scheme for the transfected mammalian cells what you can use. So basically you can transfect the cells with this particular look expressing vector, and it is actually going to express the luciferase. And then ultimately you lyse the cells and you test into a microtetraplate reader, and it will give you the signal for the luciferase. And this is the expression of the screening by the GFP. So if your cells are expressing the GFP, then it is actually going to give you the separate peak into the flow cytometry, and that can be used for screening of the untransformed cells versus the transformed cells. So once the screening is over, right, that you actually got the desirable recombinant clone. These clones could be of the cloning present in the cloning vector or to the expression vector. Cloning vectors are mostly being used for studying the transcription, replication, or the preparation of the genomic or the cDNA library, where the expression vectors are being used for studying the mechanism of translation or the protein productions. Now, talking about the protein production, this is general steps of protein production. So here you have the promoters, then you have the coding sequence, and then you have a termination sequence. The RNA polymerase will come and bind. It will actually going to synthesize the messenger RNAs, and then the ribosome will come and it will synthesize the protein, right? This means for the protein production, the two things are very important: the promoters, right, and how strongly it is actually going to attract the RNA polymerase and as well as the ribosomes. And based on these two components only, people have developed the different types of protein production machinery. So you can actually have the different types of host, and in all the host, you basically can have the different types of promoters. You can actually have the different types of accessory molecules so that you can be able to modulate the activity of the promoter as well. And this is the criteria to select the expression system. So it depends on what is the quantity of the protein required. It depends on the size of the protein. It depends on the source of the organism and the expression system, and it also depends on to the downstream application. So downstream application is one of the criteria which is very important when you are talking about the industrial applications. For example, if I'm trying to develop a protein for vaccine purposes, then I cannot use the E. coli as an expression system, because then it is very difficult to get the FDA approval. Because if you are using the E. coli expression system, then probably the bacteria or bacteria-derived product could be released, and it could be responsible for the allergic reactions. So E. coli expression system basically require the promoters, you require the ribosome binding site, you require the termination site, and you require the affinity tags. So this is what is shown here, right? So you have a promoter where you have the -35 regions, you have the -10 regions, and depending upon the composition of -35 region and -10 regions, the promoters could be strong promoters or could be weak promoter. So when I say strong promoter means it is actually going to facilitate the multiple rounds of RNA polymerase binding and removal. Whereas when we talk about the weak promoters, it does not facilitate the RNA polymerase binding or cycles of RNA polymerase binding. Then based on the promoters, you can have the IPTG inducible promoters, right? So you can have the LAC promoters, you can have the TAC promoters, you can have the TRIC promoters. And all these different types of promoters are present in the different types of vectors or different types of plasmids.
Sir, sorry to interrupt, sir, but it's can you please just wind up in because the next speaker is waiting to enter.
Okay.
Sorry, sir.
Yeah. Okay. So expression of a fusion protein in E. coli is require the easy purifications. It requires the targeting of the cellular components, and it also depends on the half life of the protein and the solubility. So there are multiple criteria and how you can be able to choose a particular expression vector or expression system in the E. coli. And then you will also have the different types of fusion tags, what you can use for easy purification of the proteins in the E. coli. And this is the scheme of purifying the fusion protein in E. coli, and so on. So with this, we can conclude the molecular cloning part, right? And what we have discussed about the how you can be able to clone the protein into an expression system and how you can be able to perform the expression. So if we have very quickly, if we have one or two questions, I will be happy to answer.
Do anybody have any questions? Please ask, sir, because we are going to wind up. No questions, sir. Thank you, sir, for your lucid explanation on cloning, and that too in a very stepwise manner, answering all the hows and whys. And we are sorry that we couldn't give you more
No,
time.
it's fine.
Yeah, the next speaker is
completely understand.
waiting, sir.
Yeah, sure. It's not an issue.
Sure.
Anyway, I will share the presentation with you. So you
Yes,
can
sir.
share with
That
the
would be
participants
helpful.
in case because they missed out many of the things, right? So they can go through with the material, right?
Yes, sir. That would be helpful. Thank you so much, sir.
Okay. Bye. Thank you.
Bye, sir. The participants are, please wait for a few minutes so we can start our next session. Thank you.
Sir, welcome, sir. Welcome, Dr. Chakrabarti. Could you start? Welcome, Dr. Chakrabarti.
Yeah. Can you hear me? Am I audible?
Yes, you're audible. Okay, let me start. Okay.
Okay,
let me introduce you first, sir. Huh?
Yeah.
Welcome you all, participants and also Dr. Chakrabarti. Today we have with us Professor Dr. Chakrabarti, a distinguished figure in the field of CTVS. CTVS means is cardiothoracic vascular surgery. Dr. Chakrabarti currently serves as a professor and head at the Department of CTVS at KPC Medical College, Kolkata. He is also associated with Narayana Hospital, Fortis Healthcare, Apollo Gynecology, Kolkata, and as a lead thoracic surgeon. He is one of India's pioneer thoracic surgeon and also performed first-time VATS and RATS-enabled lobectomy of lung and robotic assisted thoracic surgery in Eastern India. He has more than 20 years of experience in surgical discipline and has his area of special special interest include lung cancer, RATS, VATS, lung volume reduction surgery, tracheal reconstruction, and lung transplant. He has published more than 30 research papers in journals of national and international repute. He has also trained more than 20 postdocs in the thoracic surgery discipline. Today, Dr. Chakraborty will take us to a fascinating journey of cardiothoracic robotic surgery, art, science, and ethical considerations. Dr. Chakraborty, you may please start now.
Yeah. Thank you for the kind introduction. Good afternoon, everybody. Today, for the next 80 to 90 minutes, maybe 1.5 hours, we'd like to share our experience of journey in cardiothoracic robotic surgery, art, science, and ethical considerations. Am I audible to all of you? And can you see the screen?
Yes, I am audible.
Yes, sir.
You can
Yes,
see
sir.
the
You are
screen.
audible and visible.
Okay, thank you. So this is my disclosure. And the journey of robotic thoracic surgery cannot be discussed without this understanding that the journey of human evolution is impossible without the evolution of hand. And as we all know, that instruments are the extension of hand. So to understand the science of robotic surgery, we have to understand the function of hand. And this hand, this evolution of hand has made us a human being. From ape to human, this evolution is always associated with the development of our movement of fingers, which involves the movement of our thumbs along with the opponents. That is opponent's movement of fingers. Now, Aristotle and Newton, you know better than me. Now, Aristotle and Newton, why I am presenting here? Because Aristotle was one of the famous philosophers, and whose understanding was there is no need to understand the laws of nature because it is always inbuilt and we cannot do anything out of it. And it was Isaac Newton who actually found out the laws of nature. And actually, what he did, the revolutionary part of this is that in between God and nature, he brought the human being, understanding the laws of nature. So this is a wonderful journey of our human scientific development. Now, as I have told you, that hand and next comes the instrument and then comes the robots. And these all robots or the instruments, they are all the extension of human hand, essentially extension of human hand. So you can find on the left-hand side of the screen how we are plucking, how you are getting the fruits with the help of the robots. And on the right-hand side, the first one is the conventional surgery. We are working with the hand, and then comes the thoracoscopic surgery, and the lowermost, the robotic. This is Karel Kopek. Karel Kopek was the first, he's a playwright, Czechoslovakian playwright, and actually this Rosam's Universal Robots display, here we first find the name of robot. Robot in Czech means slave labor. And actually it was his brother Joseph who actually coined the term robota. Robota is robot, slave labor. And this is Isaac Asimov. Robot, the understanding of robot did not emerge from a scientific laboratory. It emerged from fiction and playwrights understanding. Isaac Asimov, biochemist who worked on tyrosines and was a professor in biochemistry in Boston University. And he has plenty of fiction, and it was he who actually popularized this understanding of robot in his fiction writings. And this is his important quote. It is change, continuing change, inevitable change, that is the dominant factor in society today. Even today, when we are sitting trying to understand in this continuing education, this we have to remember, that no sensible decision can be made any longer without taking into account not only the world as it is, but the world as it will be. This is very important, the world as it will be. Now, this is the basic three rule of robotics. Before we go switch over to our journey of cardiothoracic, this is very important understanding that the three rule of robotics, and this is also not developed by a scientist. It is developed in a fiction story. That is one first rule number one. Robot may not injure a human through his action and allow one to cause harm. Rule 2 is robot must obey all the orders given to it from humans, and only where the exception is where such orders would contradict the first law. If there is any step to harm human, the second rule is it won't follow. And the third rule is a robot must protect its own existence except when to do so would contradict the first law or the second law. So this is the three rule of robotics, and it is still pertinent in our practice as well. Now, robot is an extension of hand, and the movement is intuitive, it is automotive and actual. And the hand is mimicked by the robotic arm. So this is the brain. On the left hand side, that is the master, and on the right hand side is the robot we commonly call, where the hands are there. The right hand side screen, that is the slave. Now, what are the advantages of robot? Before going into the advantage and disadvantage, first thing we should talk about what is a robot. A robot is a reprogrammable, computer-controlled mechanical device equipped with sensors and actuators. And the advantages, from the surgeon side, there are plenty of advantages: versatility of tasks, experience, hand-eye coordination, dexterity, and quickly can process extensive, diverse, qualitative information. But there are drawbacks as well: tremors, fatigue, imprecision, ineffective at sub-millimeter scale. Several drawbacks are there. And the advantage of robots are, repeatability, their stability, diverse sensors, tolerant of ionizing radiation, manage multiple simultaneous tasks, many such. Disadvantages are it's large, expensive, cumbersome, not so versatile, and technology is developing, still in its infancy. So this is Charles Devil. Charles Devil is the father of robot who developed the industrial robot in 40s and known as the father of robot. And he developed a magnetic process controller that could be used to manage these first robotic machines. As computer technology began, in 1954, Devol patented the first manipulator with playback memory. So this is Charles Devol. And this is Joseph Engelberger. And this Joseph Engelberger first developed the company for industrial robots. So this is a brief history of journey of robots in industry. Now, this is very important. Characterization of a robot. And these robots, there are different types of robots with 3 discrete categories. In one place, the robots have a passive role to play. There, the role of the robot is limited in scope and its involvement is largely low risk, particularly in our domain. Even in industry also, the role of robot with limited scope when the robot has a passive role. Restricted role, this is another role. The robot is responsible for more invasive tasks with high risk, but is still restricted from essential portion of the procedure. The essential portion of the procedure is done by the human being. So this is the restricted role. And the third one is active role, where the robot is intimately involved in the procedure and carries high responsibility and risk. This comes in the automotive industry nowadays. Now, this robot characterization is very important, and understanding of this robot characterization. So when you evaluate a robot, we have to see all these points: degrees of freedom, workspace. Each has its own meaning, workspace and resolution, be it in a cardiothoracic surgery robot or in an industry or wherever it is. This mechanism type, inertia stiffness, speed force, dynamic range, bandwidth, all these points are very important. Like, what is degree of freedom? In next few slides, we'll like to explain all this, but let me talk about few words. Like, degree of freedom is an important concept in robotics. It defines the number of independent motions of which a robot is capable. So independent motions, that is the degree of freedom. And workspace is loosely defined as all of the space that an end effector can reach. That means a robotic hand can reach to this space. So this is a simple concept. And mechanism type, it depends upon 2 traditional broad categories, serial and parallel. I am not going into the detail, but basically the major difference is that whereas serial linkage have a large workspace, parallel linkage is stiffer because it has multiple links supporting each joint. So this is the problem in parallel. So comes the inertia and stiffness. Inertia and stiffness are both related to the material property, and the inertia of a robot is determined by its size and the density of its material. So these are the points by which you can characterize a robot. Whichever company they are coming from, you have to assess these criteria, these properties. And in addition, a greater inertia of robot makes safety a more critical concern because the robot will gain kinetic energy as it moves. So this is very important. Similarly, back drivability, when we are talking about, is essential in case of a surgeon. He ever want to move the robot by hand while it is unpowered. When there is a power loss, you have to move the robot. So this is the back drivability. And The gearing of a transmission might take 50 revolutions of the motor to result in only a tiny motion of the end effector. So this is the speed and force relation. Similarly, there are points of dynamic range, force control versus position control, and I'm not going into this. But most important one part is bandwidth. Bandwidth of the robot is very important because when you are working in a telerobotics, the bandwidth work is very important because if our input signal is given to the robot to move back and forth very rapidly, the robot will attempt to execute this instruction and move at a desired rate. As the input frequency is increased, the robot eventually will not be able to keep up due to its limits in stiffness and inertia. So that stiffness and inertia will prevent its movement which one is expecting. And this limiting frequency is known as bandwidth. So this is very important while doing a robotic surgery or any robotic activity, particularly from a distance in a telerobotic way. So these are the robot characterizations. Now, the left-hand side, this is the degree of movement. Roll, yaw, pitch, coming forward, backward, right, left, up, down, rolling. And this is the workspace. And these are the different issues which I am talking about, the stiffness, inertia, blah, blah. So these are the movements, the force. This you have to think about. Using a force while drilling, while grazing. So precision is very important. Very, very important. This is an interesting clip I would like to share. This is a OT staff who was working in a night duty and suddenly he gets into a war where he finds something interesting. And he will find, what is it? Just have a look for a couple of minutes. This is the OT, what is working, and he suddenly looks at the instruments, different instruments, and looks at the robotic hands. And he finds that each and every movement of his hand.
Sorry, sir, to disturb you. The voice is not clear. It's cracking.
Can you hear me now? Hello? Excuse me.
Is it my problem?
Sir, the video cannot take the load of the bandwidth. Sorry.
Okay.
Okay. So that's why your audio is disrupting. Huh?
Okay. Just listen to my words. This is OT staff who was working inside a robotic OT theater in a night, and suddenly he was working with all his movements of his hands in a free. When we are talking of the degree of freedom, you will find that how a robotic hand mimics almost all the movements of hand. Just an interesting video you may like.
Sir, I want to tell you that you can put off your video and keep running the video of your slide. Then only then you can manage, I think.
Okay. All right. My video is off.
Yes, sir. Yes, sir. Yes.
Now you can see the video.
Yeah, it's loading. It's loading. It's loading now. Let me see. Yeah. Yes. We can see. We can see.
হাইফাই জিনিস আগের জন্য খুব রোবটের সার্জারি সব দেখিয়েছি
Am I audible now?
Yes, sir. Yes, sir. Yes, sir.
Yes, sir. Audible.
Yes, sir, audible, sir.
So you now can find that there was no autonomy of that robotic hand. It was a surgeon who was inside the console who was actually operating and almost mimicking all the movements of that dancing operating room staff. And this is the degree of movement you can find that from the console, from master side, you can work, you can stitch, you can, and this is the movement of the fingers you can see with the hand is doing with all the precision, the stitching is going on. So this is amount of the degree of movement which you do not find in laparoscopic instrument, and that is the extension, and that's why it is told that is minimal access surgery, laparoscopic surgery or thoracoscopic surgery is transitional from hand to robotics. So these are the types of robots in healthcare: the assistive robots, medical robots, and humanoid robots. These are the assistive robots like wearable robots, rehabilitation device, surgical robots, prosthesis. So these are the areas where we can work. These are wearable robots, helping, assisting in movements and for taking some materials. And these are the medical robots. On the left-hand side, you find the surgical robots, the hospital robots, exoskeletons, or the care robots, caregiving robots. On the right, lower, caregiving robots. And these are the humanoid robots. And you are all aware of the name of Sophia, world's first humanoid robot. So robot has multiple uses, even in the medical as well as in the industrial world. So these are the key companies in the healthcare robotics market. Huge, and lot of them are coming up. So this is very important, the robotic surgery precision. This is the precision at which the tremor is lost. Like, to that extent, a convincing illustration of a low, how human and robots can work together to improve surgery. Just look at it, how it is being peeled with this minimum movement. Similarly, in retinal repair, in eye surgery, a retinal surgery requires precise positioning of a laser within 25 micrometer of a target. And in order to avoid the damaging blood vessel, if a retinal vessel is damaged, a retinal hematoma and subsequent blindness can occur. So this much of precision we need. The unaided human hand cannot reliably direct a surgical instrument to within less than 100 micron of its target. Furthermore, as the surgeon becomes fatigued and intention tremor develops, that further decreases accuracy. So this much precision we need, and that is helped by a robot. And intention tremor decreases accuracy. Finally, the eye itself has a natural motion of 200 Hertz and acts as a moving target. So the combination of these factors create an operative situation that lacks the precision needed, but is well within the capabilities of current robotic technology. So this much of precision is needed, and it is helped by a robot. Now these are the different generation of robot. These are Da Vinci robot. I'll come into the other robots as well. From 1990 to this DV5, the latest one, which has a haptic feedback that gives the almost similar feedback a hand can have about the feeling of a texture of a material. We are mostly using Da Vinci XI and Da Vinci X. Da Vinci SI and these models are now backdated. Mostly in our country, Da Vinci XI and Da Vinci X model is used. These are the other robots. SSI Mantra is from India, and Varsius minimally invasive robotic system. This is the Corindus of Siemens. This is used in the cath lab, that is for angiography and angioplasty. And these are different robotic system you can see. So it is not only a single, multiple, at least 8 to 10 robotic system has come up in the medical field for the surgery, maybe more. And nowadays, a lot of them are coming from China, Korea, Japan, apart from America and Europe. This Surui robotic system has been developed in China. It is a uniportal, the C, the right hand side. It is a single port with a multiple snake-like hands, which can work like all the robots which I have seen earlier. This is very interesting. This is trauma pod, and this is a semi-automated tele-robotic surgical system. Like a trauma patient, earlier which I was talking about, is that someone is in the bedside of a patient, and the robotic surgeon is working from a console away from the patient. Here, there is no human being. Only surgical robots are there who will be operating on a patient. And this is a semi-automated tele-robotic surgical system which are used in military setting and for managing the trauma patient. But this must be a low risk. This is very interesting. India's first robotic system performed cardiac surgery over a distance of 286 from Gurugram to Rajasthan. Again, surgery has been done from Gurugram to Hyderabad. So this is a tele-robotic system. Tele-surgery is done from hundreds of kilometers away. Now let's come to the art of robotic surgery. There are types of robotic cardiac surgery. Almost all cardiac surgery, thoracic surgery, which can be done with the help of hand, can be done with the help of robot. All the valve surgeries, coronary artery bypass grafting, congenital heart surgery, even robotic heart transplant also done. So now you look at this video. কোথায় বাবা চলছিল না মনোযোগ করে দিয়ে গেছে এটা অফ করে একটা ভিডিওটা অফ আছে করা যাবে ভিডিও অফ তাহলে করে দে চালু করে Now, on the right hand side, you can see we are doing a vascular surgery with the robotic anesthesia with a minimum incision. So these are the precision which can do with the robots. And the approaches of robotic thoracic surgery, maybe you can see on the chest, the C mark is the camera. We go to the 8mm camera, and R1 and R2 are the hands of the robot, and A is the assistant port. And approaches of robotic surgery may be to the multi-port or two-port, even single port. We call it uniportal. So just imagine, earlier with the 20 centimeter, 25 centimeter, we used to operate on lung or heart. Now it is 8mm, 3 or 4 ports, 8mm or 10mm, 3 or 4 ports, even with a one port that is about 4 centimeter. These are the advantages of minimal access thoracic surgery. The types of thoracic surgery, apart from the cardiac, we can go with the lung resection, lung cancer, any tumor inside the chest wall. Robotic thoracic surgery can do with the excision of tumor in the windpipe, in the diaphragm, even with the foot pipe, esophagus. All surgeries which where you can think about, imagine about the heart or lung, can be done with the help of robot. And these are some of my video clippings. You'll find that we are operating, excising a diseased part of the lung or on the posterior wall of the chest. And these are the instruments which actually I am operating from a console. And what I am moving with the help of the hand, and that is being replicated, and intuitive movement is there, and we are removing the tumors or removing a lung diseased lung. So this can be done with a robot. Earlier we used to do it with the help of the hand. These are some other clippings, but we are reporting in a TB lung. You know, in the tuberculous lung, particularly in our country, we have plenty where patient coughs out blood and the lung is densely adherent to the chest wall, and we are operating on it with the help of this robotic hand with a small incision, so there will be less pain, recovery will be much better. And on the right hand side and left hand side upwards, what you are seeing is the stapler. This is also another development in the robotic surgery that helps in the stitching and cutting of the tissue. This robotic stapler has come within the last 3 years. Earlier it was not available. So this is the uniportal robot, and we are doing here. In India, we are also doing this uniportal robot, that is robotic surgery with one small incision, nothing extra, 3 to 4 centimeter incision. And there are papers coming up with the Indian experience. This is a fantastic surgery. What you are seeing, we are stitching a windpipe which had a tumor, and that is the trachea and bronchia, and we are saving the lung. Otherwise, earlier we used to remove the whole of the lung. Now we have removed only the tumor, and this is being done with the help of robot. And this is the bipolar grasper of the robot, and the right hand side, it is the needle holder. And on the lower down, what you are seeing, we are operating on diaphragm. What is diaphragm? Diaphragm is the partition between the chest and the abdomen. And if a diseased diaphragm, what happens that sometimes stomach and everything gets up inside the chest, and we have respiratory distress. So this is the diaphragmatic surgery which we are doing with the help of robot. Now, what I told earlier, that there are risks and every procedure must be safe. So there may be some trouble like this, which I have faced. By operating, suddenly there's a bleeding, but we have to manage it. The rule is, don't hurry, stop, give a pressure, be prepared, take control. So this is the rule. And this also can be done, which we can do with the hand. We can do it with nicely with the help of robot with proper training of the skill. So this is the state of art surgery. Even the transplant, robotic lung transplant, you just imagine that robotic lung transplant can be done with the help of a 3-inch incision or a 2.5-inch incision. And it is being successfully done all over the world. Not many centers, but several centers are doing this robotic lung transplant. This is a paper from New York University where Dr. Stephanie has presented in last STS meeting of doing a fully robotic double lung transplant. Stephanie from NIV. So these are animation, just putting this way, and this is how the lung is placed inside. Now, area. The concept of robots, you know, 10 to the power minus 9.
Sir, you are not audible.
Can you hear?
No, sir, we can't hear you.
Hello? Can you see the video?
Yes, sir.
Okay.
Sir, Dr. Chakrabarti, are you here?
Yeah, tell me.
Yeah, you're audible now. Audible now. Yeah.
Yeah. So this is another aspect, because the limitation of robot, particularly in our surgery, is there is no haptic feedback. That is, the robot machines, instruments which are working, we don't know what is the texture, how much pressure we are giving to the tissue. And this is the way the research is on to understand the tactile response. So these are the research going on, and you see the palpation experiment with the Da Vinci robot. Da Vinci DV5 has come, and this is the way a slave is following the master. You look at the right side, it is a master who is working from the console, and trying to detect the orientation of the embedded stick. The servo motors are moving the platform, and with each movement, the user's finger, you will find. In our models, which we are working in XI or X, there is no haptic feedback. We have to look to the screen and find the visual clue and have an idea how much pressure we are giving and what is the texture of the structure. From the visual clue, we have to understand. But now in this DB5, after all this research work, there is a haptic feedback, and the master, who is the sergeant who is working in the console, can understand how much pressure is being exerted and what is the texture, the context of the texture. Here you see that in the normal, it is not moving, so they do not get any haptic feedback. No haptic feedback. These are the different strategies to understand the texture and having the feedback. The pressure strategy and dragging. So from this, we'd like to switch over to our understanding of robotic surgery, that it is demonstrating potential to enhance surgical precision. There is less patient morbidity and improve the overall outcomes. And the intraoperative complications, catastrophes, are equivalent to the open surgeries or laparoscopic or thoracoscopic surgery. But the limitation which I have told you earlier is a lack of haptic feedback. And definitely cost effectiveness are frequently debated issue because the cost is higher. Now let's come to the most important question, that is ethics. Ethics of this robotic platform, ethics of robotic surgery. And this is very important and less frequently discussed. Now, the advantage of robotic surgery as a part of minimal access surgery, that the both patient and organizational advantage of this minimally access surgery over traditional open surgery. It has a less postoperative pain, less scarring, less stay in the hospital, quicker recovery, small incision, less stress on immune system, many things, many advantages. And there are multiple papers coming out, the systematic review coming out, and at least it has been proved there are some advantages. And when comparing with the thoracoscopic surgery, these are non-inferior till date and superior to the open surgery. Now comes the principle of ethics. This is very important. This is very important in our surgical domain as well as in the industrial domain in the use of robot. And the first point is, in ethics, it is the autonomy, then comes beneficence, then comes non-maleficence and justice. These 4 points are very important ethical questions which we have to talk about as a teacher, as a researcher, as an implanter, as a performer. These 4 points of ethics principle we have to abide. First is autonomy, that is acknowledge the people's right to make choices for themselves based on their own values and beliefs. Their choice, it's not our choice. Patient's choice. One ought to prevent and remove evil or harm. One ought to do and promote good. That is the beneficence. And not to inflict evil or harm. That is non-maleficence. And the justice is treat others equally and fairly. So these are the points. Beneficence is acting in the best interest of the patient. Autonomy is respecting the patient's right to make their own decisions. Non-maleficence is not causing any harm to the patient. And justice is the concept of emphasizing equality and distribution of resources among individuals. This is very important. This justice and autonomy, these two are very important concepts, and that should never be forgotten in the field of robotic surgical practice, medical practice, and even in any social practice of robot. Now, the beneficence and non-maleficence. The beneficence and non-maleficence, the primary goal is to benefit the subject of the treatment. No surgery should ever be done for the sole benefit of a surgeon, health system, or the patient's family. This is very important. This number 2, that surgeon feels that doing a robotic surgery will be beneficial or helpful for the surgeon, technically helpful, economically helpful, whatever it is, this is not done. Or the health system, that is the corporate hospital, or the patient's family, no. Patient is the sole decisive person about this. And some studies demonstrated benefits of robotic thoracic surgery over VATS, including shorter length of stay, improved limb of dissection, lower conversion to open rate, whatever. This is the beneficence of the patient. That is very important. This is autonomy. This is very important. Many a times, many places, this robotic use are promoted by manufacturers and hospitals to elevate the benefits and hid the negative facts from patients on their websites and media. This is encroachment of the autonomy. These obscured facts degrades the ability for self-determination. So, guiding indications for operations and surgical judgment still supersedes patient autonomy. So, autonomy is very important, and justice is offering robotic surgery as a guise for superior technology. Robot is a superior technology. Superior technology is not equal to justice, and maybe a representation of the standard of justice. Not necessarily a superior technology. Because it is superior technology, it should not be practiced. This is a wrong understanding of robotic practice. Now, this is very important. That is a major concern with autonomous surgical robotics, the responsibility, liability, accountability, and culpability. What is responsibility? All force appears to be similarly sounded, and sometimes we understand responsibility, liability, accountability, all are same. No. Responsibility is duty to deal with something, duty to complete a task. This is responsibility. What is liability? Liability is a present obligation to pay or fulfill a duty to another party arising from a past event. This is liability. Fulfill a duty to another party. And accountability is, so liability and responsibility is different. Accountability, what is accountability? Accountability is obligation to be answerable for the result of the task. Obligation, you are accountable, means your obligation to be answerable. And what is culpability? Is a state of guilt. So each point of autonomous surgical robot, so when we are going from a manually operated robot to highly automated robot. These concerns are very important. Who will be responsible? Who will be liable? Who will be accountable? And who will be culpable? Where surgical error occurs, where there is any shutdown and problem related to the robot occurs. So, till now, there is no suitable framework to deal with this. So, till date, we are guided by these levels of autonomous driving, that is from the Society of Automotive Engineers, their taxonomy and definition of terms of driving automation system for on-road motor vehicles. So from this, we are discussing till date, even in our robotic surgical understanding, robotic surgical fields. So like zero automation, the driver has full control of the driving task, and level 5 is full automation. The vehicle performs all driving tasks under all conditions. And the driver assistance is level 1, partial automation is 2, and conditional automation is the vehicle can control most of the driving tasks. So just imagine from 0 to 5. And so from the ethical point of view, just I'm going to the last slide. So who will be responsible for level 3? Who will be responsible for level 5? Who will be liable? Who will be accountable? Who will be culpable? With the development of autonomous robots, humanoid robots, this question is cropping up. And this is yet to be resolved. Because should an autonomous robot be deployed and make a poor decision that leads to a preventable medical harm? This question has to be answered. Because when we're talking about the semi-automated robotic, we are doing tele-robotic trauma surgeries. Who is responsible? Who is culpable? Where does the chain of responsibility begin? Is it the manufacturer? Is it the hospital or owner? Is it the surgeon? Where? From where it will start? Which level of automation? Who will be responsible? These are the ethical questions which has to be answered. And whether the human should take disproportionate burden of responsibility, because it has been studied in several research that there is a tendency to give a responsible to the surgeon or the operator the responsibility more, even in an automated surgical system. And this is known as moral crumple zone or cognitive bias. This is happening. So this has to be sorted out. So robots has come to stay, and so is the robotic thoracic surgery. And I started quotation. It is change, continuing change, inevitable change. That is a dominant factor in the society, and we have to think in terms of, not only in terms of world as it is, but the world as it will be. The science, art, and ethics of robotic thoracic surgery should be based on this perspective. Thank you. Any question? Hello?
Thank you, sir.
Yes.
Sir, I am Dr. Deblina Sharkar from Bagnan College. Sir, in one of the picture, you have shown that two robot arms and one camera arm have been inserted inside the patient's chest.
Right.
And there was another port also, assistant port. So, what is the function of that assistant port?
Actually, there are two issues. One is the more arm you use, the expenses drop up. Each of the arm, each time you use, you have to pay extra 20 to 25,000. All right. So in our country, to reduce the use of the robotic arm and the cost expenses we use. Moreover, with this assistant port, this port is little bigger, and we usually extend this port to use the suction machine and some other machines which are yet to be developed by robotics. Use like the thoracoscopic or laparoscopic instruments, and at the same time we remove the specimen, tumor, lung, whatever it is, through that code. That is the assisted mode.
Okay, sir. Thank you. And sir, there is another small question. Just a curiosity that those GI surgeries, these are also very delicate surgeries. So, are these robots? They are used in GI surgery for colon cancer, rectal cancer.
GI surgery is not a delicate surgery compared to a cardiothoracic vascular surgery. So, we are working on the coronary vessels, we are working on the heart, lung, and you have seen the vessels which we are working in the lung where I was dissecting, and there was an injury, there was a bleeding. All right. So, GI surgery, they are definitely doing it. There are multiple centers in India, even in Calcutta. There are fantastic GI robotic surgeons who are doing the pancreatic cancer surgery, lung, colon cancer surgery, stomach surgeries, whatever. Even liver resection is being done robotic in the city of Calcutta. We have fantastic robotic surgeons here.
Okay, sir. Thank you. Thank you.
Yes, Dr. Namrata Rai, do you have a question?
Yes.
Yes.
Good evening, sir, and thank you very much for a very captivating lecture. What I have no expertise in this field. Just out of curiosity, I wanted to know that you have mentioned that the approaches can be uniportal, biportal, or multiportal. So,
Yes.
sir, when it is uniportal, which basically means one incision is made, does that imply that only one robotic hand will operate there, or that is enough for two robotic hands? Or I mean, how do you decide how many ports are required? That is what I wanted to ask, sir. Thank you.
Even in a uniportal robot, we can use 3 to 4 arms, mostly 3 arms, from different angles. So it is usually when we go for a multiportal robot, our incision size is 8 mm to 12 mm. But for a uniportal, it becomes 3 to 4 centimeter. So in 3 to 4 centimeter, we can use, because most of the robotic arms are 8 mm or 12 mm. If when you are 12 mm. Otherwise, these are all 8 mm. So in a 3 to 4 centimeter long incision, we can use 3 to 4 hands of robot.
Thank you, sir.
In the Surui system, which the China has admitted, you find the snake-like arms. It is coming out from one end only.
Right. Okay, sir. Thank you so much.
Well, is there any question from our audience, our participants?
Yes, sir. I have a question. Good evening, sir. My name is Dr. Arunima Datta. I wanted to ask that before this invention of this cardiothoracic robotic surgery, there was a indication that patients who were to undergo the surgery for opening the thoracic cage or invasive surgery of any kind, there was a condition called medical counseling. So, is it difficult? It has become more difficult to convince the patients and their the patient those who are looking after the patients, to convince them about the robotic surgery, or they are more keen on going on for this? No, you just go on for invasive surgery, open the body, and that will be more acceptable, because although we are talking about robotic surgery, more precision, more taking away the after effects more effectively, but in our culture we consider the doctor as next to god. So if doctor is doing the surgery, he is there, he will take care of any kind of situation that is coming so far. Whatever the conditions may be, the doctor will take care of it. How much it is important to convey this message that robotic surgery is more effective in place of this open invasive surgery? How much it has been difficult for the medical counseling part, for the patients and their family?
Thank you for the question. A society who has a God also has a devil. So, whatever it is, God may be, a doctor may be a God, at the same time may be a devil. The question which has to be answered is the question of informed consent. Informed consent and autonomy of a patient is right, because open surgery is also safe. When you are offering a robotic surgery or minimally access surgery, because it is a superior technology, that doesn't make any sense that it has to be carried out. Whether it is beneficial for the patient, this is more important, and superior to the other methods. Like when we are talking about robotic surgery and open surgery, there are multiple papers, studies, systematic review, meta-analysis that it is superior to open surgery. But till date, there is no adequate data to prove that thoracoscopic surgery or VATS surgery and robotic surgery, the robotic surgery is superior. There are papers coming up, but still the meta-analysis, systematic review, randomized control trial are still going on. So there is no conclusive evidence that robotic surgery is far more superior to thoracoscopic surgery or laparoscopic surgery. This is most important. This information has to be given, but the reaction of the patient and patient relatives are something different. Because few people think superior technology is equal to better for the patient. It's not always, it does not go parallel, but it is non-inferior. Definitely it is non-inferior. It has been claimed to have less pain, small incision, early recovery, but still it has to be systematic study has to come up to prove this. And other way is there is a patient and patient relatives are scared that some machine is operating, so something worse may happen, bad may happen. So patients, the reactions are two types. One is very much receptive, for that insist to go for a surgeon even with a 12 centimeter size tumor. You have to bring it out. You cannot bring it out with a 1 centimeter incision or 3 centimeter incision. Still patient relatives go for the microsurgery and robotic. This is one extreme. And another extreme is, no, actually the machine will operate, anything can happen. If the machine crashes, what will happen? They don't understand this. There are all safety measures taken for all these issues. Am I clear?
Yes, sir. Thank you, sir, for the detailed inscription. But personally, I feel that in the context of today's scenario, people are very scared of machines. Although there are experts and everything, they will consider that no, sir, you won't be there. Or for tele surgeries, that the doctor is not there. He's controlling it from the different part of the country. Then how will it happen? So we think, basically, I think that there needs to be more counseling sessions and more productive approach towards convincing the people, those who are going into surgery or the family members, that what are the benefits of going on to a robotic surgery over an invasive surgery.
Let me clear one misunderstanding.
Yes, sir.
That is, even in a telerobotic surgery or even in a robotic surgery in a hospital, there is always a bedside qualified surgeon by the side of the patient. Never ever there is a surgery, telerobotic surgery is done, that someone staying in 280 kilometer away and there is no surgeon. These surgeons are also equipped in operating any form of complications with the bedside. This is a mandatory and this is an ethical issue. This cannot be done.
Yes, sir, that is that I have understood. But have you ever faced such kind of a situation that where you had difficulty to convince the patient or the family members that it will be better for a minimal invasive surgery over an intensive open
Oh
thoracic?
yes, oh yes. To make strong understanding, we have to convince them, clarify them, but patient's autonomy is very important.
Yes,
Okay,
sir.
sir.
Thank you.
Any other question? Please continue. Huh? Anybody there to raise question? Hello. Okay, sir. It's time for vote of thanks. Uh, we, from the part of organizing committee and the participants, thank you for your illuminating lectures that spans from history of robot to robotic surgery, advancement in robotic surgery to the ethical considerations that arises out of this technological advancement. Thank you very much, sir, for your kind presence in this occasion. Thank you. For the participants, I have an announcement that assistant coordinators have a few words with you, and so please wait for a few minutes, participants.
Hello. Good evening, everyone. Once again.
Good evening, ma'am.
Yes.
Good evening, ma'am.
Good evening, ma'am.
Good evening, ma'am.
Hope you're enjoying the
Good
lectures.
evening, ma'am.
Yes, ma'am.
Yes, ma'am.
Good
Yes,
evening,
ma'am.
ma'am.
Yes, ma'am. They are very versatile.
Thank you. We have tried our best on our part. Now, in the inaugural session or on the first day, we missed a few of the participants. I will again try to interact with them if they are present. Mrs. Pooja Srivastav, if you are here, please. Mr. Shobhan Rai, Mr. Vignesh Karthikeyan, Dr. Sudeshna Nandi, Mr. M.R. Ramesh, Dr. Shagota Pal, Mrs. Rintu Matthew, Dr. Anikul Islam, Dr. Kumud Chaudhary. Anyway, maybe better luck next time. So now regarding tomorrow, tomorrow is an important day for the participants because we will have the first PowerPoint presentation in the morning session right from 10:30 a.m. One thing is that a few 5 or 6 of the participants will give their presentation, but the rest of the participants must also join at 10:30 a.m. and they must also be part of the presentation, which will be given by 6 of the participants first. And in an update, uh, uh, we would like to inform all the participants that regarding the number of slides, we have revised a bit the content slides. That means there will be a slide for with the title the name, the affiliation, the introduction slide, and there is usually a last slide that is given, thank you slide. Excluding those, that is 2 slides, minus introduction and thank you slide, the maximum number of content slides can be 8. We have revised it. It can be 8 maximum, 5 to 8 slides per participant. And there will be experts also during the session, and accordingly we have conveyed to the experts that the number of slides can be maximum 8. That is the content only. Okay, content slides. Hope everyone is noting it down. Again, uh, the moderator who will conduct the session will again tell you the rules and regulations once again tomorrow. And in the last session tomorrow is the MCQ test 1. So the moderator who will be present during MCQ test 1, he will read out the rules and regulations of MCQ test 1 during that session. So now, is there any question?
Excuse me, madam.
Yes.
One thing I wanted to know, what will be the exact presentation time, and will there be any question answer sessions followed by the presentation?
Yes. The time for presentation, please note everyone, is 10 minutes plus 5 minutes for interaction, max 5 minutes for interaction. Okay, so the presentation time is 10 minutes plus 5 minutes for interaction. So in no case should it exceed 15 minutes total time. Okay.
Thank you, madam.
So again, I am summing it up for the participants' presentation. Number of content slides, max 8. It can vary from like 5 to 8, anywhere in between. If your content is not for 8 slides, you can make 5 slides, no problem. But max 8, excluding introduction and conclusion. That is thank you, excluding introduction and thank you. And the time for presentation is 10 minutes plus 5 minutes for interaction. So with this, I think we will conclude today's session. And lastly, I would like to wish all the participants who are presenting tomorrow best of luck, best wishes, and best of luck to all the participants for MCQ test 1.
Thank you.
Yes, best of luck.
Thank you, ma'am.